Hello all, I am Vaishnavi Churi. I am from Yash Vidya Niketan, Virar East. I am your science teacher. I am going to teach you science part 2. Today we are starting with our first lesson number 1, heredity and evolution.
Hello students. Today we are starting with Science 2, chapter number 1, Heredity and Evolution. Now about heredity, we have studied in 9th standard, that is heredity and variation. I hope you all remember what is heredity. So what is heredity? Heredity is nothing but the transfer of biological characters from one generation to the other. So when we talk about heredity, it is a transfer of genetic characters or biological characters, that is through the genes. So genes are transferred, genetic characters are transferred from parental generation to the next generation. When we talk about genes, what is this genes? What are this genetic material? So these are nothing but the DNA. You must have understood, you must have learned in the previous standard what is DNA, that is deoxyribonucleic acid, and what is RNA. Hope you all remember it. So Mr. John Gregor Mendel is called as father of genetics or pioneer of the modern genetics. He is the one who discovered the phenomena of genetics. He had done a lot of research, lot of work in the genetics. After Mr. John Gregor Mendel, in 1901, the reason behind sudden changes that were understood by Hugo de Vries in his mutational theory. What Hugo de Vries understood that there can be sudden changes in the combinations, sudden changes in the genes, and this is called mutations. Mutations are nothing but the sudden changes in the genes. This theory was proposed by Hugo de Vries. After Hugo de Vries, in next year 1902, Walter and Sutton, these two scientists discovered that chromosomes are present in the pairs when they studied the cells of grasshopper. Until then, it was not known to anyone that there are chromosomes which are actually the genetic material. So this discovery was done by two scientists named Walter and Sutton. A few years later, in 1961, Francois Jacob and Jacques Monod they discovered the process of protein synthesis occurring with the help of DNA in bacterial cells. What is protein synthesis? When the body needs any protein, these proteins are synthesized through the DNA. They are synthesized by the DNA, and this discovery was a great discovery done by Francois Jacob and Jacques Monod. This led to the development in genetic engineering. Now, these two scientists, when they discovered that proteins are synthesized by DNA, this process where it helped in the other further discoveries in genetic engineering. We'll move to the next important topic, that is transcription, translation and translocation. When we talk about protein synthesis, so whenever body needs any protein, the body gives that information to the DNA. So information about protein synthesis is stored in a DNA, and this synthesis of appropriate proteins as per the requirement is necessary for the body. So this information which is stored in DNA is through this information these proteins are synthesized by DNA through the mRNA. Okay, this process is called central dogma. So central dogma is nothing but the proteins are synthesized by DNA through RNA. This process is central dogma. Now this in this central dogma, this mRNA plays an important role. In this central dogma, the mRNA plays this important role. mRNA, which is produced as per the sequence of nucleotides on the DNA, only one of the two strands of DNA is used in this process. You all must be knowing that DNA is a double helix structure. So one strand out of this two double helix structure of DNA is used in this process of transcription. Now, whatever information about protein synthesis is present in the DNA, one strand of the DNA is used for the further protein synthesis process. Now, mRNA is present in the nucleus of the cell. It forms the complementary sequence as per the coded in the DNA. So the sequence of nucleotides in mRNA being produced is always complementary to the DNA strand used for the synthesis. Now here you can see this DNA strand, which is a double helix structure. One of the strand is used. Accordingly, the codes as per the nucleotides, as per the uh, purines and pyrimidines are present on the DNA strands, the mRNA strand is formed using the complementary code to the DNA strands. Now, besides, you must be knowing that DNA, it has adenine, guanine, cytosine and thymine, while RNA, instead of thymine, the uracil is present. So besides, there is uracil in RNA instead of thymine. So whenever this complementary coding is done, when the mRNA is formed in the nucleus, this process of RNA synthesis is called transcription. Okay, so in this transcription, mRNA is formed with the complementary codes by the DNA. This process is occurring in the nucleus. Now, once this coded message is, it forms the complementary sequence in mRNA. Now, this mRNA, this transcription process is done, and then this mRNA comes out from the nucleus. Now, this mRNA you can see here, it comes out from the nucleus, it comes into the cytoplasm. Now, it brings in the coded message from the DNA. DNA, it already had some coded message. It formed the mRNA. Transcription process is done. Once this transcription is done, this mRNA comes out from the nucleus, and the mRNA message contains the codes for amino acids. This code for each amino acid consists of 3 nucleotides. It has the code, the one code has the 3 nucleotides, and that is called triplet codon. Now here you can see this mRNA, it has triplet codons. A group of 3 nucleotide is the one codon, that is called triplet codon. So this mRNA can have the codes for the amino acids. Now, there are so many amino acids required by the body, present in the body, and these codons were discovered by Dr. Hargobind Khurana. He is a scientist of Indian origin, and he had also been awarded by Nobel Prize in 1968 for his discovery of these codons for 20 amino acids. He had discovered codons for 20 amino acids, and he had been awarded by Nobel Prize. Dr. Hargobind Khurana. Okay, now this mRNA, it got the triplet codons, right? Transcription process is done. Now mRNA is out in the cytoplasm. When this mRNA comes in the cytoplasm, it has the codes by the amino acids. Now, these amino acids are supplied by the tRNA, right? tRNA is the transfer RNA. Now, this is happening in the cytoplasm. So, this tRNA, it supplies whatever amino acids are required by this mRNA, right? Messenger RNA gives him the information that I want such amino acids. These amino acids are provided by tRNA. Now for this purpose, tRNA has anticodon having complementary sequence to the codon on mRNA. Now mRNA is having some codes, triplet codon. Now tRNA provides this amino acid with the anticodon, and this process is called translation. Transcription was DNA to mRNA. It was in nucleus. Now mRNA to the tRNA. The tRNA gives the anticodon is called translation. This is happening in the cytoplasm. Now the amino acids brought in by the tRNA. tRNA has given this anticodons. Now they are bonded together by the peptide bond. You can see here this is the translation, that is tRNA providing the anticodon, that is a triplet codon, right? And this ribosomes, there are ribosomes keeps on moving from one end of the mRNA to the other end by the triplet codon. It will move from one end to the other end by the distance of one triplet codon, and this process is called translocation. So ribosomes moving from one end to the other end by the distance of one triplet codon is called as translocation. Okay, so I hope you all have understood what is transcription, what is translation, and what is transcription. Okay, now we have seen what is transcription. Now translation is done in the cytoplasm, then translocation is also done, it's completed in the cytoplasm itself. Now there are sometimes some changes, as I told you, Hugo Devries, when we talk about mutational theory. Now what is mutations? Mutations are nothing but the changes, the sudden changes in the sequence of the genes. So sometimes any nucleotide of the gene changes its positions. These positions are changed by any of the reason, and that causes the minor change in the sequence is called mutations. Now mutations can have the good effect as well as the bad effect. Okay, so in case of some species it can have the favorable effects, favorable changes, but sometimes due to this mutation, due to this sudden changes in genes, the person can have any disorders or any genetic diseases. Okay, so when we talk about mutation, we have to remember the name of the scientist who proposed this mutational theory is Hugo de Vries. Okay, so here we complete the heredity and hereditary changes, that is, we have studied about transcription, translation, translocation, and again the one important topic, that is a mutation. Moving to the next part of the lesson, that is evolution. Now you must have heard about evolution from your lower standards. You have studied evolution in history part in the lower standard, how the human being is evolved from his ancestors. So you must be knowing that we are evolved from the monkeys, from the apes, right? That is the basic information we already have studied, but here we are going to see how the evolution has occurred in the on the earth. You must be knowing that evolution, the life started on the earth from the primitive cell, that is a one single cell, right? You know that there are some unicellular organisms, there are some multicellular organisms. So unicellular organisms started existing on the earth, and then it uh, it it formed the complex organisms, complex compounds, and the multicellular organisms started to exist, right? We will see in detail in the later part. But basically, what do you mean by evolution? Now, evolution, when we talk about, evolution is nothing but it is a formation of new species due to the changes in specific characters of several generations of living organisms as a response to natural selection. Whatever the new species are forming, that are forming with new changes, and that is also they are these effects are also the response to the natural selection is called evolution. Now, evolution is nothing but is a gradual change. It is a slow process. It is the changes are occurring but very slowly. It is occurring at slow pace. Gradually, it is changing, and it takes a long period. Okay, so evolutions means the gradual development, gradual change occurring in living organisms over the long period. Now, this is very slow process through the development of organism, through which the development of organism is achieved. Now, when we talk about evolution, human evolution, we know that we are evolved from the apes or the monkeys. So we can see the difference between the monkey and ourselves. So we can find that there are changes, but these changes have occurred over the long period. So gradually development, gradual changes occurring in living organisms over the long period is called evolution. Now, when we talk about evolution, uh, we know that around 3.5 billion years ago, life had been non-existent on the earth. You know that earth was a just hot sphere, right? Hot gas sphere. There was no existence of any life on the earth around 3.5 billion years ago, right? After that, once the temperature got cooled, once there were some changes in the environment, in the temperature, overall biosphere was formed. Then the first primitive type of cells, that is the we say the pre-cells, right? So first primitive type of cells may have been formed from the mixture of different types of organic and inorganic compounds. So when we talk about the first the life which started existing on the earth, that was the first primitive cell which formed by the mixture of organic and inorganic compounds. Now, there may have been some differences among those cells. According to the principle of natural selection, some may have shown good growth and some may have perished, which could not adjust with the surrounding. You all must be knowing about the survival of the fittest. We are going to study about the Darwin's theory in the later part. But when we talk about survival of fittest, what do you mean by survival of fittest? Survival of fittest is nothing but on the one who will survive, can fit who can fit to the external challenges or whatever the uh, conditions are there. So the one who will fit to these challenges, who will fit to the external conditions, that organism or that living organism can only exist, right? So this while these primitive cells will start existing, from unicellular to multicellular, different animals started form this new species started forming, and they started existing. But after some time also, there were some animals who could adjust with the changes in the environment, changes in the surrounding, and they could sustain, while others got perish. Now you must be knowing the example of dinosaur. Dinosaur we have seen the movies, right? Not in actual. But when we talk about dinosaur now, we don't find any dinosaur now. Why it happened so? Because they could not survive to the conditions which had occurred, which had which was challenging for them when there was a sudden environmental changes occurred on the earth. You must be knowing that there is a story, there is a theory which states that there was a meteoroid which crashed on the earth, and due to that there is a huge sand dust clouds were formed, and because of that the the herbs, the plants, they could not, the sunlight was blocked, and because of that the plants got died. And after that, the herbivores who are dependent on the plants, they also started perishing, they also started dying. And after herbivores comes is the next trophic level, that is carnivores, and so on. This food chain got stopped, and then the animals got perished, extincted.